So let's look at the actions of the antimicrobial drugs in a little bit more detail. Inhibitors of cell wall synthesis are very effective because bacterial cell walls are made of peptidoglycan, and these drugs will target the peptidoglycan. They don't hurt human cells because human cells don't have cell walls. Also, any eukaryotic cells that do have cell walls, such as plants, do not have cell walls made up of peptidoglycan. When these cell walls are interrupted, their synthesis isn't able to complete, the cell walls become weak, and then the cell can undergo lysis, and all of the insides of the cell will basically explode to the outside, and the cell dies. These drugs, however, are only effective on actively growing bacterial cells because they do not hurt a cell wall after it's been synthesized. They simply prevent synthesis from being completed. So if a bacterial infection is not growing, if the cells are not replicating and making new cells, then these drugs will not be effective. Inhibition of protein synthesis is the second mode of action that a lot of antimicrobial drugs will use. Protein synthesis obviously is a common feature of all cells, both prokaryotic and eukaryotic, so one might think that it wouldn't be a very good drug to have toxic selectivity. However, the ribosomes, as you recall, are involved in the translation, and eukaryotic ribosomes are much different from prokaryotic ribosomes. So these drugs will target specifically the prokaryotic ribosome. That is why they don't hurt our own cells. Injury to the plasma membrane is typically used for antifungals. Eukaryotic membranes typically have what we call sterols inside of them to give them support. However, the fungal cells have a different type of sterol than what human cells have. Therefore, these drugs can target specifically the sterol that is unique to fungus cells and not those that are unique to human cells, and therefore the human cells are going to be just fine while the fungal cells are going to be attacked. Inhibitors of nucleic acid synthesis typically act as competitive inhibitors. They're shaped like the substrate that is usually involved in nucleic acid synthesis. Because they're shaped like that particular substrate, they block the real substrate from binding to the enzyme and allowing the reaction to occur. An example of this is PABA. PABA is a substrate for an enzymatic reaction that is necessary for folic acid, which eventually plays into nucleic acid synthesis in microorganisms. However, a drug that is shaped like the PABA molecule will bind to the enzyme and won't let the actual PABA bind to it, therefore the nucleic acid synthesis can't occur. It's important to know, however, that drugs that competitively inhibit DNA polymerase or RNA polymerase is that they can actually affect host cell DNA and cause mutations. However, they're not really considered to be too dangerous because this is rare. They also can be used not only for bacteria but also for viruses. And of course, since they inhibit nucleic acid synthesis, that stops protein synthesis before it can even happen. So they interfere with protein synthesis. They are used against viral infections. However, they do have the downfall of possibly affecting host cell DNA and causing mutations, but because of the risk versus benefit, they're not considered too dangerous to be used. Inhibitors of nucleic acid synthesis work very much the same way that competitive inhibitors of the synthesis of essential metabolites function. In fact, PABA would probably be classified more as a competitive inhibitor of the synthesis of essential metabolites. But either way, if it is an inhibitor of DNA or RNA synthesis, it's preventing that from being synthesized. If it is an inhibitor of synthesis of essential metabolites, it's preventing those metabolites from being synthesized, which are necessary for cell growth, cell reproduction, and cell function. But both of them are going to act similar to competitive inhibition. Now we're going to switch to a different topic, and I will go ahead and end this particular recording here, and we'll start up on the third section.